to tonight's Phi Kappa Phi faculty lecture. I'm Jeremy Jackson, Professor of Economics in the Department of Agribusiness and Applied Economics, Director of the Center for the Study of Public Choice and Private Enterprise, and also President of Phi Kappa Phi. We also have a board of officers, which include Dennis Cooley, our treasurer, Elizabeth Crawford Jackson as secretary, Suzanne Kelly as past president, and Jim Deal as past secretary. Phi Kappa Phi is not a fraternity or sorority, it is an honor society. Membership is not granted to all, but must be earned. Phi Kappa Phi members at North Dakota State University truly are among the brightest and highest achieving students in the nation. And at a university known for its tough standards, NDSU students invited to join at this level of academic performance typically represent about 3% of the university's student population. Invitation is an honor that gives you not only the right to wear a special recognition ribbon during graduation, but your achievement becomes part of your permanent transcript. It is the only academic honor society eligible for that designation at NDSU. In the past, the Phi Kappa Phi lecture has been preceded by the annual initiation ceremony, but because of the current pandemic environment, we are not holding a formal initiation ceremony this year. But I do want to say congratulations to all of this year's Phi Kappa Phi inductees. The Phi Kappa Phi lecture was established in 2015 by NDSU President Dean Bresciani to recognize faculty whose outstanding high-impact research, scholarship, or creative activity consistently supports NDSU's top-tier research status, and who have a proven ability to effectively present their research to a broad community in the land-grant tradition. Each of our 3 finalists for our 6th annual Phi Kappa Phi faculty lectureship receive a modest financial award and, of course, our admiration. And I want to congratulate the finalists who were considered for the award this year, including Dr. Bill Wilson, Distinguished Professor of Agribusiness and Applied Economics, Dr. Tom DeSutter, Professor and Soil Science Program Leader, who gave the Fall 2020 Phi Kappa Phi lecture, and Dr. Kalpana Katti, Distinguished Professor of Civil and Environmental Engineering, about whom you will soon learn some more. I also want to thank Jean and Bill Green, our Vice Provost for Faculty Affairs and Equity, who facilitated the selection process and made arrangements for tonight's presentation. After Dr. Katti's presentation, Dean Bresciani, President of North Dakota State University, will offer some brief remarks. And now please welcome Dr. Mike Kessler, Dean of Engineering, to introduce Dr. Katti, who will then present the Phi Kappa Phi lecture for this year.
Thank you, Jeremy, and thank you everyone for coming out tonight. It's my pleasure to introduce Dr. Kalpana Katti. Dr. Katti is a University Distinguished Professor in the Department of Civil and Environmental Engineering here at NDSU. She's also affiliated with the Materials and Nanotechnology and the Biomedical Engineering programs. She is a Husky. She got her PhD from the University of Washington in Material Science and Engineering. And here at NDSU, she's been really active in establishing state-of-the-art materials characterization labs, tissue engineering labs, and really successful in bringing federal grants for a wide range of research. And she's also spearheaded the doctoral program in materials and nanotechnology at NDSU, and has led a team of researchers in the establishment of a Center for Engineered Cancer Test Beds at NDSU, and serves as its director, and also serves as the NDSU science lead for a NSF-funded $20 million EPSCoR Center for Biointerfaces in Science and Engineering. She's authored over 200 publications and has received awards from a number of places, including the Journal of Colloids and Interfaces. She's a NSF Career Award grantee, a fellow of the American Institute for Biological and Biomedical Engineering, and just a number of other awards, but I don't want to take up all of her time. So Kalpana, I'll turn it over to you. Congratulations.
Thank you, Dean Kessler. About 400 years ago lived a certain gentleman in the city of Pisa. He was a great thinker and engineer who built his own telescope and collected a lot of data. And through the power of his amazing data and his amazing mind, he professed that the earth moves. And as a matter of fact, he was the first individual.
You're on mute.
Okay, can you hear me now?
Yes.
Sorry about that. Okay, I'll start again. So about 400 years ago, there lived a certain gentleman in the city of Pisa who was a great thinker and also a great engineer who built his own telescope and was able to collect a lot of data with that telescope. And through his tremendous amount of data and his great mind, he was able to profess that the earth moves. He was the first individual, at least in the Western world, across the Himalayas, to say that the earth moves. He said that with great conviction and also tinged with a bit of arrogance. Because of that, he faced the Great Inquisition. He faced the Great Inquisition that also delayed the Western world from understanding that indeed the earth moves. Then about a few hundred miles north and about 200 years later came another individual, a great writer whose prose actually read like poetry. And he understood the problem. That is, it wasn't so much what Galileo said, and it wasn't so much the problem with his data, but the problem was how he said it. How he said it was a problem. And that's what he said. If he said it nicely, the Inquisition would have left him alone, and also the world would have understood sooner that the earth moves. So the thing that needs to be said today is the statistics of breast cancer and prostate cancer. If you look at the incidences worldwide of the various cancers, breast cancer and prostate cancer are right at the top, and the blue bars are the numbers of incidences per year, and the red bars are mortalities. So basically, we are looking at a very large number of individuals, 1 million deaths occurring annually each year due to prostate or breast cancer alone. So when it comes to prostate or breast cancer, we're looking at each year, every single year is like a COVID year. I would like to let that sink in a little bit, that every year for prostate and breast cancer is indeed a COVID year. So if you look at how prolific this cancer is, we're looking at about 1 in 8 women and 1 in 7 men that actually are likely to get breast cancer or prostate cancer in their lifetime. And the incidences of deaths occur because of a process called metastasis, when the cancer transports to a distant location in the body. So the question to ask is, should cancer be deadly? I'm Kalpana Katti from the College of Engineering, Department of Civil Environmental Engineering at NDSU, and it is my great honor that the Phi Kappa Phi Awards Committee thought of me worthy of presenting today's lecture. So I'm immensely grateful to them for this honor. I would also like to thank my colleague in the department, Ravi Yalavajula, for nominating me for this award, our department chair, Dr. Stewart, Dean Kessler, Provost Fitzgerald, and President Brashani for the tremendous support to this NDSU family. I'd also like to thank my family. I'd like to thank my beautiful, smart, and forever pragmatic daughter, Priya, who's always the voice of reason for me. I'd like to thank my husband, who's a collaborator who gives numbers to all of the words. I'd like to thank him for his extreme pervasive nerdiness. He's the only person I know who can recite the value of pi to the 15th decimal place. I'd like to thank all of you for joining in from across the world, scientists, researchers, and collaborators. Thank you very much. And last of all, I would like to thank an individual who's not going to join over Zoom. His name is Dr. Suresh Shastri, mathematician, also my dad, who passed away suddenly and quietly in his home just last month. I thank him for instilling me the ability to think beyond the mundane events of life, which is often a predicament for my gender. So I thank him for inspiring me to think of greater things in life. So the question to ask is, why should cancer be deadly? But really, in order to understand that, we should look at why is cancer deadly? So all of those deaths occur due to a process of metastasis when the cancer actually transports to a distant location, which for prostate cancer and breast cancer happens to be at the bone site. So these cancers, when they transport to the bone site, the cancer is terminal. And anti-cancer drugs are often ineffective in that situation because a lot of those drugs are really designed for the primary site. The animal models fail. The animals die before metastasis to bone happens because there's no intervention done. There's no chemotherapy or radiation done. So the animals die. So we don't have animal models. We also don't have human patient samples, at least not enough, because by then the patients are often in a hospice and it's hard to obtain samples. So our thought process was to come up with an engineering test bed of cancer, kind of borrowing that term from engineering of like design of products, like there's a test bed for the Model X car of Tesla. So in the same way, what if we had a test bed of cancer on which we could test various different aspects of the disease and come up with personalized and appropriate therapies? So in order to do that, we're going to have to first create bone. So since this cancer is metastasizing to bone, both breast cancer and prostate cancer, we first need to generate bone, for which we use a technology which was first proposed about 30 years ago by the researchers Langer and Vacanti in Boston. And this is called as tissue engineering, which is really a simple idea. You take cells from the individual, you let the cells grow and proliferate in the lab and take those cells, patient's own cells, and seed them onto a structure called as a scaffold, which is a mesh-like structure made up of engineered materials. It's porous and it's degradable. So the cells are now fooled into thinking they're inside the body, and they start growing tissue in the shape of the scaffold while the scaffold degrades. So we have been rather successful in being able to make bone using this method of tissue engineering. As a matter of fact, we can create the bone that is chemically, biologically, and mechanically identical to human bone. As a matter of fact, even the process by which the bone mineralization happens in this scaffold, in this engineered system, is identical to human bone. Okay, so now we've created the bone, which is just like human bone, and then we take human cancer cells, in this case, prostate cancer cells. We take these human cancer cells and we seed them onto these bone scaffolds, and we are able to generate these tumors. So what you're looking at is the first ever in vitro, that means in a Petri dish, model of human prostate cancer that has metastasized to bone. So the prostate cancer is at the bone site, and it is the first ever such model. Here's some more pictures of the prostate cancer at the bone site. So if you look at a simple cartoon of what is really happening in metastasis, so the cells are growing very nicely at the primary site, and they grow and proliferate. And at this point, the anti-cancer drugs try and kill it. All the early detection methods are in place. So for most patients, we try and kill the cancer at this point. But in a few patients, a few of the cancer cells dissociate from the tumor, lose adhesion, and enter the bloodstream, and they swim far away. And they swim far away, especially to the bone site. So they go to the bone site and they colonize again at the bone site, and they start growing again. Now, the cancer that is growing at the bone site is still prostate cancer, but it is genetically altered. It's a little bit of a different animal at that point. And that is why the anti-cancer drugs don't work. So what we did is, if you look at the primary tumor, it has a very specific genetic makeup, and it has a very specific genetic identity. So we can analyze the tumor that we actually made, and we are able to find that indeed what we made is that late stage bone metastasis prostate cancer. So that is indeed the case. We did the exact same thing with breast cancer. So what you're looking at is the first in vitro model of breast cancer that is at the stage of bone metastasis. So here's some photos of this bone metastasis breast cancer. And now we can extract the tumor literally on a daily basis. So we can extract the tumor on a daily basis, and that is something you cannot do from patients. So we're able to do that with the test bed. So I'm going to show you hard evidence and data that shows three things that we can do with this test bed, and three applications in general. In the first case, if you really look at why is it those bone metastasis, breast cancer, and prostate cancer patients die, it's because they really die because of skeletal failures. They literally, their bones break. So we can use the test bed to evaluate how various different cancer types influence the bone. So we can do that. We can also look at markers. For instance, right now, there is a test that you can get for prostate cancer, but there is no test right now for bone metastasis of the cancer. So we can use the test bed to discover new markers for metastasis, so whether there will be metastasis or not. And the third thing we can use the test bed for is to use it to screen new drugs that are under clinical trials, or new compounds that are not even drugs yet. And that would come up with, that would help us come up with new therapies that are appropriate for the individual patients. So these are the three things we can do. Let's look at the first one about the impact at the bone site. So I'm going to show you both breast cancer and prostate cancer. So let's first look at breast cancer. So we looked at two particular kinds of different kinds of breast cancer. We looked at this one that is called MCF7, which is a low metastasis breast cancer. And then we're going to look at another one, which is this MDA231, which is the high metastasis. This is called as the bad triple negative breast cancer. So we look at both of them, and now we generate these tumors at the bone site in the test bed. And if you look at the pictures, they look very different. In the first case, the low metastasis makes these dense tumors you can see over here. And the other case, which is a triple negative, it does not make those dense tumors. So morphologically, when the cancer arrives at the bone site, depending on the type of cancer, it's actually looking quite different. So why is it that the cancer cells are sort of moving through the blood and going straight to the bone? Somehow they're sucked into the bone site. So we set up a simple rig with which we can actually measure how much of the cancer cells are actually migrating to the bone site. The deeper blue represents more cells have come, less blue means less number of cells have come. And basically we can see both kinds of breast cancer do like to be sucked up close to the bone site, but one is more migratory than the other. That happens to be the highly metastatic one. So that tends to go more to the bone site than the other, and that is actually the more dangerous one. So now we look at how this breast cancer, when it has traveled and arrived at the bone site, how did it influence the bone? Because bones are breaking. Remember, that is what is happening to the patients. So much of work is done in trying to understand the cancer, but really the problem is that their bones are breaking. So let's just look at the fundamental way in which bone growth is happening and whether cancer cells, when they arrive, do they do something? Well, again, we used the same two different kinds of breast cancer, the low metastatic and the high metastatic. And there's actually a very complex biochemical machinery that exists that is like business as usual for the bone growth to happen. That is called as the osteogenesis pathways, osteo as in bone and the growth of bone. So what we find that both of these cancer cells, when they arrive at the bone site, what we're noticing is they're whispering something to the bone, adjacent bone cells. And that which they whisper seems to completely disrupt the growth of bone. Okay, so lots of data here again, but that biochemical pathway is called as Wnt beta catenin signaling. Okay, so that's an important machinery that needs to go on as usual for normal osteogenesis to happen. Well, what is happening is that these cancer cells, when they arrive at the bone site, disrupt that machinery. I just want you to pay attention to the fact that the black bars here are the normal bone, and the other two bars are the two different kinds of cancer cells. And you notice one is increasing, or one is higher than normal, and the other is lower than normal. So what is happening is neither is keeping business as usual. Both of them are disrupting osteogenesis, one increasing it and one decreasing it. And both are actually harmful, and both are going to cause disruption in the mechanism. And actually, if you go deep into it, it's really a simple idea. These cancer cells have what are called as stoppers and enablers. One of these guys is a stopper. It's called DKK1. And one type of breast cancer has a lot of this stopper. And once that stopper is in, osteogenesis doesn't happen properly. And the other, when the enabler is present, it seems to exceed the osteogenesis more than normal. So both of those events are dangerous. So I think what is important to understand is what I've shown is that breast cancer cells, no matter what type, when they arrive at the bone site, they've disrupted the bone, and that's why the bones are breaking. So now let's see if this happens with prostate cancer. So that was breast cancer, because remember, prostate cancer also migrates to the bone site. So if you look at prostate cancer, again, we're going to look at two different types of prostate cancer. One is PCA and one is PC3. And PCA is the more PCA is the less metastatic one, and PC3 is the high metastatic one. Okay, so now if you look over here at the different colors, the first one here, that is your healthy bone. This is how the bone is normally. And now we look at the bone that is next to the tumor when the PCA type prostate cancer arrives close to the bone. And when the PCA type arrives close to the bone, this red color is reduced. But when the other prostate cancer arrives, the red color is increased. So again, this, by the way, this red color is a measure of the bone mineral. So the bone mineral in one case is reduced and the other case is enhanced. And in neither case, it is the same as what it should be, which is the healthy bone. So both have disrupted this mineral formation of the bone. Now, human bone has two components. One is the mineral, the calcium mineral, and the other is a protein called as collagen. So we saw that prostate cancer cells arriving at the bone site basically disrupted the mineral formation, either increased it or decreased it. We see the same thing happen with collagen. So the red color is collagen. The first one here is healthy bone. This is how we want the healthy bone to be when the cancer cells arrive. Well, turns out the PCA type tumor, you hardly see any red over there, right? But now look at the PC3 tumor, it almost looks like the collagen has gone berserk and it has become too much more excessive. And at the bottom, you see pictures, electron microscope pictures of the collagen fibrils that have gone berserk just adjacent to the tumor. So the tumor cells, as a tumor, that is not just the only problem, but they have forever disrupted the bone in the patient. So here's another picture of just basically saying the same thing, that bone formation is severely disrupted at bone metastasis due to the arrival of the tumor. So just like we saw with breast cancer, the prostate cancer also, the two different types, when they arrive at the bone, the tumors that they make are actually morphologically quite different. One is tight and dense tumor, that's actually the low metastasis kind, and the other is this sort of disorganized array. So the tumor is very different when it is at the bone site. And with the test bed, we're able to analyze that. And remember those collagen, the bone that is sitting adjacent to the tumor when it arrives, that went berserk. Here's a little photo of that in a close-up. Well, in order to say that it has gone berserk, we need to compare it to what it looks like when it's normal. So normal or healthy bone looks like this over here. And if you, I hope you can see, it's nice long striations with these black and white sort of regions that are very well organized with perfect periodicity. And that's what a healthy human bone is. But if you look at the bone near the tumor, these collagen fibrils no longer have that nice organized structure. So this is forever, the cancer cells arrived and the remodeling of bone, growth of bone, everything forever is ruined. That person's bones, no wonder, are breaking at this point. So that was the impact of bones. So the second thing I'm going to talk about is the discovery of markers, because right now, like I said, there is no test for metastasis. So we sought out to look at two different ways in which we can discover these. The first one, we call it spectroscopy based. It's really simple. You make the tumor, you pick it out from the test bed. So you literally lift it out and you pass infrared light through it. What is the purpose of that? When you send infrared light through a material, you collect a spectrum such as the one given at the bottom that has a lot of features. Each of those features come from vibrations of the bonds in that molecule that's making up that material. So a simple example is, this is a water molecule. What would a spectrum look like? Well, a water molecule has 2 hydrogens and 1 oxygen. So the different ways it can have vibration is this, it can have this stretching, or it can have another kind of stretching. So different ways in which a water molecule can vibrate, and you'll see a sort of a signature of that. Well, we get a signature of all the molecules that make up the cancer cell, and maybe we can use that as a marker for cancer if it is statistically significant and different. So that's exactly what we did. So lots of data out there. The point is, we collected the tumors over 5-day, 10-day, 15-day for 2 kinds of breast cancer at the bone site. And if you just glance at this, it doesn't look that different. They all look pretty much the same, nothing terribly different. But if you do some more detailed analysis under each and every bump, in that what you see of the features, and you delineate various different features. In particular, we're looking at the protein region, where the protein molecules are, where the fat molecules are, the lipid molecules are, where the DNA, RNA, or the genetic material is. There is lots of analysis you can do. And you can do that analysis, and the long story short is that the data that we collect over 5-day, 10-day, 15-day, 20 days of the growth of tumor at metastasis is statistically significant and different. What that means is that we can use that data to use that data as a measure of the metastasis progression. Why is that important? It's important because here in this case, we actually lifted the tumor out, stuck it in the infrared spectrometer, and collected data. We don't have to do that with patients because this whole infrared experiment can be done with fiber optics. So no biopsy needed. So that would be the future of development of new sensors for metastasis. So that is one example. The other one is very hardcore engineering based. Literally, we measure the softness, hardness, and squishiness of that cancer tumor, literally quantitatively as a measure of metastasis. So that's a novel idea. And the idea of the health of a cell being represented by its mechanical property has been shown before. For the first time, it was shown for malarial cells by Subra Suresh and Ming Dao about 30 years ago. But what we decided to do is not just to distinguish between healthy cell and cancer cell, but the idea is, can we look at it in more detail and use that mechanics as actually quantitatively a measure of the metastasis potential of that cell? And why do we think we should do that? Well, if you look at the pictures of these tumors, that's what these are. The ones on the left, the blue dots, those are just your individual prostate cancer cells, but the other two pictures on the right are tumors, and the different colors are the different structural members of the cell, parts of the cell that contribute most to its mechanical property. Those are the beams and columns of the cell that have been tagged, and we can actually see them using microscopy techniques. So what we did is we first did experiments, and we actually saw that during metastasis, the inside members are actually moving and changing and altering. So obviously the mechanical property of that cell is actually possibly changing. So we decided, and here's another photo, such a beautiful photo of prostate cancer, if you can call prostate cancer beautiful. And here's a little cartoon that shows the different structural members inside the cell. They're called as cytoskeleton. So, so these are the parts of the cell we are interested in. And now we use an instrument called as a nano indenter, and literally poke into the cancer cell, poke into the cell, and literally measure how much the cancer cell gives. So y-axis is how much force you apply, and x-axis is how much the cancer gives. So you apply the load and release it. Just that one simple experiment. And from that, we can measure the mechanical property of the cancer cell. So let's first see whether the cancer cell in the tumor is different or is it the same. So prostate cancer cells, when they undergo metastasis, so if you compare just the individual cell to the cell that forms the tumor, so you extract the tumor and indent the tumor, we see that the cell actually was stiffer by itself and became softer. And you can see over here, it became softer. So this is the mechanical property over here. This is single cell, and this is the tumor. It's actually has a lower elastic modulus, a lower mechanical property when it forms a tumoroid. So cancer cell actually became softer, squishier, and more deformable when it became a tumoroid, okay? Not only that, how much force it took. So we penetrated the same amount into the cell and saw how much force can it actually take. And turns out the maximum force is also reduced. So now we can now extract the tumoroids on a daily basis. That's kind of the beauty of the test bed, right? So we extract the tumors and now we indent them. And what we see is over the progression of metastasis, also the cells are becoming softer and softer and softer. So 5 day, 10 day, 20 days, it became more and more soft and more deformable as the metastasis process progressed. So this is the first sort of a very accurate nanomechanical measurement over the progression of metastasis. Again, just like before, that maximum force, how much can the cell take? That maximum force is also decreased on the progression of metastasis. Other interesting thing we measured is what we call plasticity. So when you load it and you unload it, how much did it get back to how it was before? Okay, so the more plastic it is, it doesn't recover. So that means it has less recovery. What we're seeing is when this metastasis is progressing, it's actually not only just becoming softer, it's becoming less and less recoverable. So it's having more plasticity in it. That's for a material scientist, that's kind of very interesting thought. What kind of material is this? And I was asking myself, what kind of material is this? Until my collaborator Dinesh, who's a mechanician, said, we're not indenting a material, we're indenting a structure. It's inside guts. Beams and columns are actually moving. And that's kind of why it has this unusual combination of mechanical properties. Nonetheless, we're able to measure mechanical properties as a measure of metastasis in this. And now let's look at inside the cell while we're measuring the properties. What is it that is making it go soft? So why is the cell becoming softer as we go? So the top row is basically individual cancer cell. Okay, and the red guys and the green guys are those structural members, those that give it its mechanical property, the inside guts of the cell or the cytoskeletal members. Now, if you look at the cell in the tumor, these set of pictures over here. Right away, you can see the red guy over here, which was all over the cell, and a lot of it is now significantly reduced. Is significantly reduced, and not only that, it has moved to the edges of the cell when it formed the tumor. You see that? Now let's look at the green guys. Those are also important for mechanics. This is the tubulin. That doesn't seem to change very much between a cell and a tumor. So, and we can sort of measure that quantitatively, basically from the same picture. So these things, the red guys or the F-actins, seem to have a big role in controlling the mechanics. So we know mechanics is changing with metastasis, and now we know why it's changing. What's causing it to change? So these F-actin, these filaments inside the spokes of the umbrella, that is the cell that is moving, changing, reducing. When all this is happening, why it's happening? Well, some genes in the cell make it happen. So we actually happen to know what genes cause those F-actins to change. We can monitor those genes, and indeed, all of them are going down. So kind of more evidence that F-actins are causing the mechanics of metastasis to change, cancer cells at metastasis to change, and that's facilitated by very specific genes. So that's what I meant by when the cancer cell arrives at bone, it is a different animal. It really is. Okay, so that was prostate cancer. Now I'm going to show you results on breast cancer, but now we do one additional experiment. Not only we apply the load like that, static, but we do a dynamic load. We do a dynamic load, and that enables us to measure what is called as the viscoelasticity of the cell. And again, the same experiment. Test bed is fantastic because we can just pick up the tumors on a daily basis, not possible from patients. And now we have two breast cancer, the same two breast cancer cells that we talked about before, the low metastatic one, the MCF7, and the high metastatic one. And this is where differences come about. The low metastatic one, no matter how much indent we give, over time, as the tumor progresses, it's becoming softer. We saw that for prostate cancer, and we're seeing the same thing with this type of breast cancer. On the other hand, the triple negative, the highly metastatic cancer, doesn't seem to do that. So it's not getting softer. It's staying pretty much the same. So it doesn't seem to be affected very much. Okay, so that's also a description of the metastasis progression, because other things are happening. And this viscoelasticity we talked about, it can be measured by something called as loss tangent. And again, we are seeing differences between the two kinds of breast cancer. And we see that the MCF7, the low metastatic one, is having much more viscoelastic characteristics. So different ways in which we are actually measuring the properties, either static or dynamic, and they're giving us a quantitative measure of the progression of metastasis to bone in particular. Now let's look at pictures inside. So when all this is happening in breast cancer, in terms of how soft it is and how squishy it is, let's look at the insides and see what is making these properties change the way they are. The one at the top is the MCF7, the low metastatic breast cancer, and the one at the bottom is the MDA231, the highly metastatic cancer. The first picture is the individual cell. The red guys or the F actins, notice, are all over the cell. And now when the tumor forms, it's much reduced. The red is much reduced, much lower F actin, and also it has gone to the edges of the cell. Well, we saw that happen in prostate cancer. But now let's look at what happens in the triple negative breast cancer. Those reds don't seem to be reduced. And that's why this ties in with the fact that our mechanical property wasn't changing so much. So not only are they going to the edges of the cell, and they're also not reducing. So just like we did with prostate cancer, we know what genes are working for these F actins, and we can watch those genes. And for the low metastatic cancer, they're all going down. So it ties in. The genes go down, the genes that cause the F actins. So the F actins go down, and so the mechanical property goes down. Well, we didn't see that mechanical property go down for the triple negative breast cancer, and the genes are also not going down. So it's all very consistent, the connection of mechanics to the biochemical mechanisms. Okay, so now something very interesting. Remember those red guys that sort of get reduced, the F actins, and they go to the edges of the cell? Well, turns out the back ends of those stick out outside the cells, and those are called E-cadrins, and that is why the cell-cell adhesion happens, and they connect. So the more F actins go to the edges of the cell, the more those back ends will stick out, and the more adhesion will happen, and the more tumor formation happens. So you remember those pictures. In one case, you had tight, dense tumoroids, and the other, you had disassociated clusters. So that's why in the first case, the F actins were sort of going to the edges, and then they were causing the cell-cell adhesion to happen. That's why they made those dense tumoroids. In the other case, they didn't. So basically, lots of these experiments I showed you that basically ascertain this very interesting new idea of using a very strong engineering mechanics approach to understanding cancer, or cellular nanomechanics as a biomarker potentially for cancer metastasis. Okay, so the third thing I mentioned I talk about is drug screening. So we can take this test bed and we can screen drugs. You don't have to do trial and error on a patient. You can do this on a test bed for personalized medicine, and you can use it to screen drugs, or rather compounds that are not even drugs. We looked at some traditional drugs, and we're also looking into some drugs that are in clinical trials. We measure what is called IC50. That means how much drug concentration is needed to kill 50% of the cells. So we're looking at a lot of that, and more recently, one interesting application has been through collaboration with Professor Shetty in the plant sciences. His group creates these very interesting extracts from specialty crops that are rhodiola, cranberry, and oregano that have been shown to have some detrimental effect on primary site breast cancer. So we wanted to see what happens to the bone metastasis breast cancer. So we took those compounds and we tested them, and our preliminary results are very promising that indeed those compounds are showing some efficacy in the bone metastasis breast cancer. And even more interesting is the impact on bone. Remember the surrounding bone that was getting disrupted? So our initial data is showing that the impact on bone is minimal. Okay, so there's a lot to cover here, a lot of work in progress, but that's indeed a very interesting direction that we want to look at. And then in general, I said that a lot of anti-cancer drugs are ineffective. They're ineffective because basically cancer cells that were responding to the drug at the primary site, when they went to the secondary site, really developed a sort of a very strong chemo resistance. And indeed, we saw that happen. Genetically, they were altered. The genes that are participating in creating that chemo resistance are enhanced. So each of these bars, you can see the black is the cells at the primary site, and the light gray bars are the cells at the bone site. And you can see they're becoming resistant. So of course the drugs are not effective. And of course, multi-drug resistance has for a long time been a big roadblock for cancer chemotherapy. So we did that, we studied that for both kinds of breast cancer in general. So overall, what I've talked about is shown you lots of data on the use of the test bed for three particular things. One is to use a test bed to truly understand the impact on bone. How is the bone regeneration, bone remodeling affected, so that we can come up with appropriate and clear therapies for each individual type of cancer within prostate cancer and breast cancer, so we can have better therapies. I also showed you the discovery of two different kinds of new markers. One is spectroscopy based and the other is engineering mechanics based. I also showed you the use of this test bed as potentially new drug screening tool with which we can screen drugs and compounds. So we can do all that. I showed you lots of data, and I think I showed you lots of evidence to show that indeed cancer should not and will not be deadly. And this is a very novel and unique engineering approach towards looking at cancer. And engineers do great things. No problem is unsurmountable for engineers. Engineers are problem solvers. We just landed a spacecraft 400 million miles away and landed it on a dime. So that was almost an unsurmountable problem. And so is cancer. So I showed you lots of data, lots of evidence that this is going to happen, that metastasis is not going to be incurable. We will have a cure for metastasis. So every year does not have to be a COVID year for breast and prostate cancer. So I showed you lots of data, and I think you'll believe me. But you know already, it's all about how it's said. It's about the synergy between the scientists, the engineers, the medical professional, the FDA, the drug companies, and the respectful conversations that happen in beautiful prose and poetry. Those conversations are going to be important before all of this science can be translated into clinical practice. So my hope is every day to learn more poetry and being able to say things to different people in different ways. So that's my goal. And here are all of the wonderful poets that participate in these conversations. Fantastic, incredible, crazy, and hardworking graduate students that have had the tremendous joy of having in my group at NDSU. So thank you. I'd be happy to answer any questions. Thank you.
Yes, thank you so much, Dr. Katti, for such a wonderful presentation, and thank you for bringing such important research to NDSU. And as Dr. Katti said, I'd be happy to help moderate. You can ask questions either if you're brave enough to unmute yourself, and if you can also show your video, but you could also put a question into the chat. I'll be watching the chat room as well, so please do go ahead and we would love to have Dr. Katti answer your questions.
I think Dr. Brashani has a question. I'm just going to give him the mic.
What was your unanticipated finding to date on the topic matter that you didn't see it coming in, the wow factor?
So I can't say the unanticipated. I mean, I can say the unanticipated, but it was not necessarily the wow. Okay. We started working on it on, actually we're working on bone. So we've been for the longest time interested in bone for the sake of regenerative medicine. And we couldn't get the human mesenchymal stem cells to grow on our material, which is a very unique nanoclay material. So we finally got it to grow, and the stem cells were able to differentiate on that nanoclay scaffold without any differentiating media. So that was very unexpected and very wow also. But coming to cancer, I remember 10 years ago in this very room, I'm so fortunate that I had the good fortune to be able to give give a lecture just very similar to this 10 years ago. And there was a surgeon in the audience who asked me, so are you sure you're making bone? What if you make cancer? And I was 10 years ago, a little bit cocky. And I said, I wish I had that problem. We couldn't get the cells to grow, and cancer is excessive growth. But after I went back and I thought, yes, so that is a problem, that cancer at bone metastasis. So the real wow in that is how quickly the tumoroids were growing, how human-like they were. So there were many wows after that. But what was not anticipated was how the stem cells were able to differentiate on this and make bone without any differentiating media. Hello? Wow. Hello?
Say, Kalpana, I have a question for you. What was it as a material science person, as somebody that came out of a PhD program, what was it that got you thinking so broadly? What got you started down this track?
So I'd have to give credit to long time ago, 100 years ago, to all my teachers and all the ones that found me annoying for asking questions. So that started a long time ago, but literally how I moved into this field of biomaterials was really a photograph and a presentation in the late 90s of what is called as the vacantee mouse. There was some group of scientists that took bovine cells and seeded them onto a shape that looked like a human ear. That was a beginning of tissue engineering. And when I saw that, it just seemed like a cool way of regenerating every single body part. And I think I was so intrigued by that. And I thought I had so much of the processing and characterization toolkit with me, I could put it to use for biomaterials for bone replacement. So I think if I can think of just one single moment, that was it. Yes.
Thank you.
There has been a question submitted in the chat room from Xiaohui Liu. Do you think that those plant extracts looking promising will work in vivo?
What we have created, the test bed, and I'm trying to say the right words so I don't offend any biologists, but our test bed is very humanoid. It is human cells. So in vivo, as in a mouse, I don't know, but it is humanoid. So our test bed is humanoid and it is very human-like. Whether in a particular mouse, there's many situations in the mouse models with bone metastasis because of the lack of intervention, the mice die. So we really don't have any good studies on that. So I believe our test bed is humanoid. So it's I don't want to say it's better than mice, but it can reduce some of the mice work certainly and speed up.
There's a question also in the chat from one of your former students. So the question is, what made the cells to differentiate without any differentiating media? How did the cells survive?
Okay, So I wish I knew who that student is, because I would then.
I can try to pronounce the
No,
name.
no. It's okay. I would have just reverted the question back to him. But short answer, we don't know. Short answer is we don't know exactly. There are some things we know about some of the pathways that are affected, but to make the long story short, we don't know fully well what it is in the nanoclays. Just want to give you a background for the rest of the audience, because it seems like who's asking the question probably knows this material system, but I just want to let everybody else know that scaffold that I described, it is made up of clays, nanoclays actually. These clays have been modified with amino acids. It's a very unique material. Amino acids, as you know, are building blocks of proteins. We use those amino acids to sort of create bone mineral, exactly like how biology makes bone mineral for making bone. So we try to duplicate that, as is a biomimetic method. But other than that, we do not know why mesenchymal stem cells differentiate. We don't know exactly. We just know they do.
Kalpana,
Yes.
I'm just curious, you collaborate with Sanford
Yes.
on the breast cancer.
Mhm.
How do you use that information for the breast cancer from the wisdom study
Okay, thank you for
to
asking.
go in with your research here?
Okay, so thank you for asking the question. I didn't get a chance to get to that, but all of the data that I showed so far is using commercial cell lines of breast cancer and prostate cancer. And as of last summer, we have an active collaboration with the oncology department at Sanford, with Dr. Gaba, and we are able to obtain samples, patient samples of breast cancer, which in collaboration with our biology researchers on campus, Dr. Jiha Kim, we are turning those tissues of patients' breast cancer into cell lines, and we're going to use those patient-derived cell lines onto our test bed. So next step in the more humanoid test bed, basically. So our plan is to do all of these experiments and show these using patient-derived cell lines. So right now, we are in the process of turning patient tissues into a cell line, turn it into something that is commercially obtained. That's what we're doing now.
President Bresciani, would you like to make some closing remarks?
Thank you. Thank you, everyone. And thank you, Dr. Katti, for an absolutely fascinating presentation that nonetheless, even a person in finance could follow. These are a bit awkward right now because obviously Dr. Dinesh Katti, Dr. Margaret Fitzgerald, and I are the only ones in the room, although we certainly have quite a few people online. But that doesn't diminish how special this recognition and presentation are, both in the quality of your scholarship, but also what it symbolizes. I think one of the most exciting things about working in a higher education environment is the opportunity to share new knowledge with our students, with the people that we serve, and to enlighten them to what's become the cutting edge knowledge in a field. But to be the one that creates that is the gift that we're all given in working in higher education, to create knowledge that no person on the face of the earth had before it was created. It is truly a gift. It's an honor. And to recognize the best of the best at one of the top 100 research universities in the nation, I wish I had the award we would normally give, but we're not supposed to be passing things back and forth by hand. But nonetheless, thank you for an absolutely fascinating presentation, but more importantly, the quality and impact of your work. You deserve our highest possible recognition and respect. And thank you very much. Thank you, everyone who's joined us online. Obviously, we would have had a full room if everybody was here. But again, that doesn't diminish the quality of what we had and got to experience this evening. And I want to thank everyone. Dr. Kessler, thank you for hosting us. And I'll turn it back to you.
Thank you so much, President Bresciani. I think that concludes tonight's program. Let's give a round of applause for Dr. Katti.
Thank you everyone for being here. I'm not sure who's controlling the screen, but I think that does it for this evening. And again, thank you very much, Kalpana.
संपलो संपलो का
Same to Kasok. Wow, amazing, amazing, amazing.
Hello. Hello,